Section 2, History of Classification. Just a couple more definitions. Species, a group of organisms that can interbreed and produce fertile offspring. A phylogeny, the evolutionary history of a species. Let's look at this phylogenetic tree, or another name is what we call a cladogram. Let me get my pen here. Okay. This is the main evolutionary line right here, with branches coming off to indicate when each of these different types of animals had evolved or developed. So if you look at this is our main branch, what came first? That's right, sharks did, then tuna, then frogs, so on and so forth. And if you notice, where did humans come in? Well, looks like we branched off of this Dimetrodon. Okay? So as you can see, a phylogeny just shows evolutionary history of when a certain species developed or evolved. Characters are inherited features that vary among a species, like locomotion, their mobility, do they have legs, they have arms, fins, wings, cilia, flagella, all of that. Another type would be your biochemical characteristics, such as your amino acids, your nucleotides, which also help to determine evolutionary relationships among species. Chromosomes structure and the number is actually one of the best ways to be able to figure out similarities between certain species. If you look at the orangutan, the gorilla, the chimp, and then the human, you can see that we have very, very similar banding structure and number of chromosomes. So, what's all this stuff mean? The greater the number of shared DNA sequences between species, the greater number of shared genes, and the greater the evidence that a species share some kind of common ancestor. And thus we can categorize or figure out where new and different species developed. The molecular clock. It is a model used to compare DNA sequences from two different species to estimate how long the species have been evolving since they diverged from a common ancestor. Okay, lots of stuff. Let's break it down. Scientists can determine how mutations have occurred, and the more mutations means the more time has passed between the divergence, or meaning the time lapse of those different types of species. So if this is the common ancestor, you can see how it has changed, and the longer it has changed, the more mutations have occurred. A few other terms. Cladistics is a system of taxonomy that reconstructs phylogenies by inferring relationships based on certain similarities. And what they use to show cladistics is a cladogram, which all of it is a branching diagram showing cladistics, but it's the exact same thing as a phylogenetic tree. They're just the same names. As you can see here, the point where they meet is what we call a node, and that just represents a common ancestor where branches originate. And then here's another bigger one. So here you can see this is where we began life, and then we evolved all the way down, and the branches of each. Some essential questions. What is a phylogeny? What are characters? How does the molecular clock function? How does a cladogram show cladistics? Some vocabulary words that you guys do need to know. Phylogeny, character, molecular clock, cladistics, and cladogram. Alright, let's go over that spider dichotomous key. So please pass out that Spider Lab. You should have 2 sheets, your answer sheet as well as the little packet of information. Please pause me while you do this. Okay. At this point in time, all of you should have your spider dichotomous keys. The background. One kind of arachnid, the wind scorpion, is so named because it seems to run as fast as the wind. The sea spider, another arachnid, lives on the ocean floor. The trapdoor spider digs a hole in the ground, covers it with a silk trapdoor, camouflages the trapdoor with leaves, and waits for its prey to walk across the trapdoor. When its prey passes above, the spider feels the vibrations and opens the trapdoor to make the capture. These arachnids are only 3 examples of the thousands of different types of arachnids. Arachnids, as well as other organisms, are often identified by their physical traits. A dichotomous key based on visible physical traits can help you identify members of a group of related organisms. Dichotomous means branching into 2 parts. As you use a dichotomous key, you read pairs of descriptions and decide which description of each pair is true about the organism you are identifying. In this laboratory, you will use and make dichotomous keys for the identification of some arachnids and some other arthropods. Alright, let's look at the anatomy for just a second. You have the crab spider versus the yellow Vajovis. If you look, you can see that both of them have what we call a cephalothorax and an abdomen. All arachnids have 2 body parts. In the crab spider, if you look at the cephalothorax, it's kind of like its head. See how it narrows down and then it kind of bulges back out for the abdomen. But if you look at the yellow Vajovis, it is the cephalothorax and abdomen is one constant straight thing. There is no narrowing between the 2. Please remember that. The other thing that we need to look at is what we call segmentation. I know this might kind of freak you out a little bit, but technically if the crab spider and the yellow Vajovis was right in front of us, if you could take your finger and run your finger along its back of the crab spider, it should be pretty smooth. But then if you run your finger along the yellow Vajovis, if you look at how its back has different kind of little plates, that's what we call segmentation, or it being segmented. You can kind of think of the blinds that are on the windows. Hopefully they are closed so you can see them, but if not, use your imagination. If you would take your finger and run your fingers down the blinds, you could feel each segment or each little blind piece of the blinds. And that's what we call segmentation. They're going to ask you about that. Okay, so number 2, use the dichotomous key on the next page to identify each arachnid. Beginning with the first pair of statements, decide which statement is true for the arachnid. Proceed to the next pair of statements as directed by the key. When the key lists the name rather than number of a statement pair, then you have identified the arachnid. Be sure to record each step of the key you use to identify the arachnid. Okay, so let's have the picture of picture A as well as the picture, the dichotomous key, the words part out in front of you. So let's do the first one together. If you look at A and then we look at the key, is the cephalothorax or abdomen segmented, or is neither one of those segmented? Again, if we ran our finger down its back, would we feel the bumps or wouldn't we? Well, you would feel them, and they'd be on the abdomen, so we need to go to where? Number 2. Does the arachnid have an abdomen with a tail, or is it an abdomen without a tail? Well, there's definitely a tail there, so where are we going? Number 3. Is the tail thick tail with stinger at the tip, or is it a slender tail without a stinger at the tip? There's definitely no stinger, so we're going to number 4. Now we got to look at the pedipalps. And the pedipalps are just kind of you can think of them like arms to help hold the prey as the spider is eating it. So the pinchers on the pedipalps with making it a large arachnid, or the pinchers on the jaws and it's a small arachnid. You got it. It is a small arachnid with pinchers on the jaws. Thus, we have identified it as a micro-whip scorpion. Go ahead and finish B through N, and don't forget to do the analysis questions. Good luck and have fun. Don't forget to complete the second part or the front part of the first journal we did, where it asked you to identify the different kingdoms with their cell type, their number of their cells, their cell walls, and to give some kind of example, as well as the 3 domains on the bottom. Thank you. Good luck on your lab.